Thank Midwestern BioAg for uh, allowing me to participate in the webinar. Hope everything's going well for everybody wherever they may be tuning in around the country. I think the first 2 weeks, if you've tuned in to uh, Edwin's webinar on carbon and John's about fertility, the desire today is to really touch on the fact that the effort and the synergy of all of that is to get those minerals, to get those nutrients that you've purchased that are inherent in your soil, and that you're growing, into a carbon biological cycle, so that they can then most effectively and efficiently be made available to this year's and subsequent year's crops. For so long, as we've all known and are gaining even further knowledge, that crop fertility has been sort of a fuel tank approach, where we sort of look at this year's crop nutritional needs, potential removal of nutrients, and we fill the tank up, thinking that ultimately we're going to then sort of get close to empty, and then fill it up again for next year. With the systems approach at Midwestern BioAg, that certainly is key with carbon, minerals, and biology. The effect really that I'm going to try and present today is about getting those nutrients in a carbon biological cycle. It really means looking at it more from a flywheel approach, managing the energy of both the microbial and plant relationship, as well as having those nutrients in a plant and microbe friendly form, as well as having the necessary biology to do the heavy lifting of converting those nutrients into plant available form. So the Edwin's talk on carbon is extremely important. We're learning more and more from the standpoint of how the soil plant system functions. That carbon is a key component, and that's definitely beneficial to have the carbon-based fertilizers, both the MBA dry carbon-based fertilizers and the liquid carbon-based fertilizers in a program, to provide that necessary platform and foundation for biology. Obviously, then having minerals, nutrients, fertilizers in a form that's not only linked and combined with the carbon, but also in a biology-friendly form. And then thirdly, the biology itself, the inherent or parent biology in your soils, are those that you may add or inoculate in that process. And one of the big drivers is that the soil biology takes up these components in a form of a carbon and nitrogen and phosphorus, for instance, ratio that they need at those bases in order to complex and build their own organisms, their own biology. So it's very clear that carbon is the driver of that engine. When we have deficiencies in carbon in the system, both long-term and short-term carbon, we'll have deficiencies in biology, and therefore have deficiencies in mineralization, of nutrients by that biology. So soil microbes eat first. Basically a review of that previous slide. They're a product of their environment. We've known that, and if it weren't for that, they would have died out long ago. So soil microbes are highly adaptive, and they're driven by their need to reproduce. In the right environment, some soil organisms can double in population, as quickly as every 20 minutes. And their need for acquiring that carbon and nitrogen in those ratios, is critical. So therefore, it's safe to assume, obviously, that soil microbes are a dependable indicator of soil health. We're seeing that more and more from the standpoint of CO2 respirations and those sorts of things. And then because of that, it's clear that carbon is the driver, of this soil nutrient microbial recycling system. I was at a conference, an acres conference, going on 10 years ago or so, and there was an EPA scientist there that made a proclamation that about 20 years from now was going to be the end of the chemical era. And that isn't to disparage the users, current or past or future of those chemicals. His proclamation was really not that agrochemicals would stop being made or that they would stop being used. His point was that they would stop working. So, using nutrient cycling and reducing our dependencies on these, chemistry measures, is also part of, what having nutrients in a carbon biological cycle does. We want to make sure that those nutrients, are in a form that the biology needs to liberate and make plant available versus the very highly soluble chemistries that can get away, quite frankly, and leak into Gulf of Mexico, groundwater, et cetera. So just a quick review on biologicals and, what is a biological anyway. There's a number of things that are coming on the market. So just a quick overview. There's probiotics, which are containing live organisms. Prebiotics, which may contain either the constituents of live organisms that were once there, but no longer containing live organisms. Biostimulants, like the LCBF, for instance, is considered a biostimulant. Biofertilizers are oftentimes, similarly referred to, as a biostimulant as well, and they really are, some of these very, very specific organisms that have been determined to contribute nutrient cycling benefits. And then there's biocontrol, biocides, very similar to products on the market, that produce induce systemic response, and help the plant from the standpoint of either defending against disease or pest, or in some instances actually having a direct effect on a pest or disease. Some of those that have been around for quite some time, so the biologicals aren't something new, but we've had BT, Milky Spore, Bavaria bassiana, Streptomycetes, Rhizobium. We all use various strains of Rhizobium on our legumes for inoculation, Trichoderma, and even human biotics, right, with penicillin, mycitricin, et cetera. So the use of biologicals, both in agriculture and beyond, is not something that's new, but it is an expanding process. So again, the probiotics, as we talked about, those are typically contain live cultures, and again, many of them are sold in those different forms, such as Trichoderma, mycorrhiza, and the Rhizobiums for peas and soybeans, vetch, alfalfas, et cetera. Biofertilizers, again, that's containing substances which contain living organisms, and applied to plant surfaces, used to colonize soil rhizosphere, both the interior and the exterior of plants. Prebiotics, some of these are even in the form of kelps, humic and fulvic substances, and they're also known as plant growth regulators, but these would come from natural sources versus synthetic sources as far as those regulators go. Biostimulant, again, sugars, molasses, dextrose powders, humates, soluble carbon products, very similar to the benefits that we see from using products like the LCBF to not only encourage and feed and then have those microorganisms that are inherent in your soil function from a nutrient cycling standpoint, but also if you're using a liquid biological as an inoculant, they would be a food source to provide the necessary energy and resources for those biologicals as well. Biocontrol, again, induce systemic response, very similar. Regalia is a perfect example. That's actually an extract, of a plant, a weed in particular, but it produces this induced systemic response that then the plant naturally builds its immune system. There's also volatile oils, cloves, neems, and those sorts of things that help primarily from insects, and prevent their it disrupts their mating cycles and even potentially, changes that. And it also has some, antifungal effects as well. Matt Kleinhens from Ohio State University has done quite a bit of research on liquid biologicals, and one of the main thrusts of his work was really to try and determine where they worked, where they didn't, and how they best need to be stacked in an agricultural system as far as with other fertilizers and those sorts of things. And he writes that the relationship between plants that produce extremely well and are healthy, and those is directly related to the robustness and the diversity of soil biology. What we've learned from scientists is that only about 1% of the known beneficial biology in the soil can actually be cultured in a lab. So when we look at biologicals, the desire is to have as many different species and so the most diversity and the most volume or biomass or robustness of that biology as well. So when you're looking at using a liquid biological or adding biology into your growing systems, this is the list that Matt at Ohio State has put together. That the diversity of the microbes in the product is key. Number one, always use those microbials with a food source, something that contains, a readily available, energy or food source for that biology. Consistency in the production of the product, stacking and having synergistic effect, with other products that it's partnered with. And then when non-performance occurs, look at the possibilities that there might be either an imbalance, a limitation on soluble carbon or carbohydrates, for that biology, or, that the plants themselves, are struggling from a nutrition standpoint and maybe not producing the root exudates that are necessary to support that biology as well. So when we take a good sound system that has a carbon included as a carbon base in fertility, in both dry and liquid form, and then we inoculate, with a broad spectrum, robust biological, we can see immediate things from the standpoint, certainly in legumes. We see growth stage advancement, we see root development enhancement, nodulation increase, are all components that we're looking for when we take the nutrients that you're already using, the management practices that you're employing from the standpoint of crop residue management and sound biologically friendly fertility. We can leverage that, by enhancing that biological community, in the planting and maintenance of a crop. This is a University of Wisconsin study that had nodulation using CX1 as liquid biological, and we saw almost a doubling, 100% increase in nodulation. Again, that's going to transform to nitrogen credits in future crop years, as well as enhanced crop yield and development during this season. The other thing is managing those nutrients in your crop residues. We're seeing really high levels of crop production. Corn crops 250 to 300 bushel. Managing those residues, just physically getting them out of your way, can be a challenge. But the other thing is to make sure that those nutrients stay in that carbon biological cycle and aren't left on the surface and oxidized back into the atmosphere. So whether it's at the beginning of the process and at the genesis of germination and seed root development, or whether it's at the end of the season and managing those residues, getting those nutrients back in to a carbon biological cycle is ultimately critical. It helps from not only a residue management and planting and field fitness the following year, but it also makes sure that those nutrients that you've bought or grown, get back into an available form more quickly. This is actually soybeans that were grown on that field after that crop residue was sprayed with LCBF and CX1, and you can see, the plants on the right are those that had the treatment. Plants on the left were the check. And again, we're getting better planting conditions, and we're cycling those nutrients back into the carbon biological cycle, for that subsequent crop in a more efficient manner. With and without liquid biologicals on corn, and obviously looking at this picture, the yield tells the story, the emergence, the lack of tip back on the corn, all those things are what we're wanting to do from the standpoint of getting those yields and getting the ROI that we need. But the other thing is that when we talk about nutrients in a carbon biological cycle, these are tissue tests of corn, where we see by supporting the plant and the fertility regime with carbon and biology, we get increased uptake of nutrients into the crop that year, which ultimately results in plant health and increased yield. The other thing is that if we're feeding that back to livestock, that corn that was in the previous couple slides back, we saw a 15% increase in protein. No difference other than broad spectrum liquid biological and support with a high quality carbon source. So the feed value is also really sort of the hidden gem. Increased yield, beneficial ROI there, and has to be there. But certainly if you're feeding it to your own livestock, or if you're able to directly market this to someone who sees and knows the benefits of those increased nutrients, crop nutrient density, which ultimately leads to animal and human health benefits, is the next frontier. And I personally am really excited to see that we're able to really move these things, in the right direction, in a very positive direction, with a sound fertility program, really good cultural practices, and supporting that systems approach with high quality carbon sources and high quality biological sources. We did this on again, demonstrating here. The whole idea is not to necessarily show all the details of the increases that we saw in amino acids and vitamins, but again, to show that by supporting and really trying to get nutrients in that carbon biological cycle, we're able to drive the nutrient benefit demands. Obviously, in a year like this, where our dairy farmers, especially here in Wisconsin, are suffering terribly from a number of factors, being able to produce higher quality forages, either on the same or less acres, or potentially be able to improve forage values and be able to potentially reduce other inputs or byproducts, is critical. And we were able to move in this particular trial, over 50% in both amino and vitamin assays, and that's critical from the standpoint of really understanding this minerals in a carbon biological cycle. Is one of the fields that we tried it on, and again, just conventional fertility, 0060, and a little bit of MAP, DAP, and I think even some urea was going on, alfalfa, if you can imagine that. And on the other side is an organic transition, and again, that's sound dry fertility from Midwestern BioAg, LCB, LCBF, carbon supporting broad spectrum biological, and we're able to get not only those increases in tonnage, but the increases in those forage quality, and move those nutrients down the chain, into that livestock, and then ultimately even into the milk or meat that livestock is producing. So again, sort of as a review, we talk a lot about mineral balancing, and there's been a tremendous amount of work over the years, and MBA was and still is a pioneer and frontier in that area of balancing minerals and understanding those ratios and balances. But I think that we're realizing that there's also the need for this balance and this natural proclivity for the plant soil system to have balance on a biological standpoint. And carbon is really the link. Carbon is the bridge, to allowing that mineral and microbial balance to occur. So just as a bit of review, and we'll post a short whiteboard of this along with the webinar, a link to it. But again, taking that the macronutrients, that Edwin talked about, amino acids being, or micronutrients, excuse me, being so critical, and we're realizing every day that there's an increasing number of micronutrients that are important, for optimal plant health, and then ultimately getting those nutrients into the livestock and human food chain is also very important. We're doing this with over 100,000, known beneficial bacteria, over 50,000 known beneficial fungi. And the key there, is carbon, ensuring that we're supporting that system if it's deficient in carbon with high quality carbon sources, like the dry carbon-based fertilizers and liquid carbon-based fertilizers, as well as then leveraging that system with, a healthy plant producing much greater volumes of photosynthesis and root exudates, so that we're pumping that liquid sunshine and those sugars down to feed that biology. And all of this synergistically builds to create the far more diverse levels of amino acids, proteins, and enzymes that ultimately results in, the quality of not only the crop from a yield perspective, but also ensuring that when we're getting optimal yields, we're also producing an extremely high nutrient density crop that stores better, a crop that comes out of the field drier and in better harvesting conditions. And so the benefits of really minerals in a carbon biological cycle is to get to that end game where we're developing strong ROIs, good strong yields, but keeping an eye on the ball relative to managing the fact that these nutrient density aspects are going to be the next opportunity in agriculture to really differentiate and maximize not only profit opportunities, but ultimately, soil health and animal and human health opportunities as well. So I think we're about the time to start opening up for questions. So first question, you noted applications with LCBF products in a conventional cropping system. Can CX1 be tank mixed with other chemical applications such as a herbicide, or will it harm the biostimulant effectiveness? We found that in most instances, as long as we stay away from anything that's real high salts, CX1 tank partners, in a variety of different ways. The biology in broad spectrum liquid biological like CX1 is in a dormant form. So it's going out, with those tank partners, and then it's not until it reaches, a hospitable environment, whether that's on a leaf surface or on the soil or both, that the activation of that biology is in effect. So the actual tank partnering and application, shouldn't be a problem. Another question here: what is TerraFed versus CX1? So TerraFed is actually the molasses-based product, the organic version of the LCBF product, and that is those sugars, carbohydrates. It's the blood of the molasses, the sugarcane plant, the molasses in particular, is the food source for CX1. CX1 is a broad spectrum biological that ultimately contains over 4,000 species of beneficial bacteria and fungi. So it's important that the combination of an LCBF product in an organic system, TerraFed in a conventional system, Boost 721-3, or those other products would be combined together in order to have the effects that you'd be desiring. Then 10 gallons per acre is a pop-up. And a follow-up to that, what is TerraFed versus CX1? Question, that same individual then had a follow-up question to that. So yeah, in a situation where you're looking at an organic application, we typically recommended about 5 gallons of each. The TerraFed and the CX1, they're seed safe. They can go in furrow. And although they wouldn't necessarily carry a large NPK number, they're gonna liberate those nutrients and work more as a bio pop-up. Protecting that seed, getting that seed off to a good start, germination, early root development, and then leveraging that through the entire season to provide the end of season results that you're looking for from crop yield and nutrient density, crop quality. Good question. I think we'll close the session now. Please make sure that if you are looking for that continuing education unit credit, that have credit for nutrient management, to email marketing@midwesternbioag.com with your name and ID number. And the deadline to submit that is next Friday, May 1st. Safe planting. Happy spring. And again, thanks for joining us today.